Okay, in this last video, we're going to finish up on how the ammonium from our body goes into the into the hepatocytes. Now, because I totally didn't screw up the last video, we know that glutamine is the primary transport of ammonium from hepatic cells into hepatocytes, okay? And once again, that's going to be done by glutamine synthetase, okay? We're going to take those glutamates that we just put through the aminotransferases, and we're going to add another ammonium via glutamine synthetase, okay? Obviously, totally didn't screw that up in the last video. We know that it's glutamine going through the blood vessels. Anyway, glutamine is now inside the hepatocytes. Now, so we need to cleave off that amide group. How are we going to do that? We're going to do that via glutaminase. Oh no, it's very complicated of a name. So it's going to take out the amide of that functional group to now make it a free-floating ammonium and glutamate. Now, once we have glutamate, we have two different enzymes it can go. It can either turn into, it can either lose its ammonium, its amide group, and just have free-floating ammonium, and that's going to be done by glutamate dehydrogenase, or it's going to transfer its amino group to an oxaloacetate via aspartate aminotransferase. Once again, that's aspartate aminotransferase. So once again, oxaloacetate is going to be aminated, and it's going to turn into aspartate. Okay. But what about those free-floating amino acids, or those free-floating ammoniums inside the mitochondria of the hepatocytes? Well, it's actually going to turn into that first kind of acetyl-CoA analog of the urea cycle, where we're going to use an enzyme called carbamoyl phosphate synthetase 1. Once again, carbamoyl synthetase, carbamoyl phosphate synthetase 1. Carbamoyl phosphate synthetase 1. Now, that's going to basically just add, that's basically going to add a CO2 via bicarbonate to that ammonium and add another phosphate. Now, if this sounds like it's going to require a lot of energy, it does. It uses 2 ATP for this, okay? 2 ATP to make carbamoyl phosphate. All right. And that's it. Next up, where I'm probably going to do, screw it, let's do glycolysis and then gluconeogenesis. And it'll be a quick round, just, we know what it is, all right?